This abstract is a little bit complicated genomic investigation that leverages the whole genome sequencing data that you cannot get in any other settings like exome or targeted sequencing. So we know that there are certain genomic events like hyperdiploid and IGH translocations that everybody keeps saying they are the initiating events. So the events that the first event. So if you have a cell acquires second-1 translocation, that cells needs that to become a myeloma at a certain point. It could be in 20 years, 40 years, 30 years, but that is the first event. That's what the current literature and everybody's going to say if you ask them this question. What we thought is that it might be there are some events before that, maybe some less specific events for myeloma, like events that can be shared also in other lymphoma that can precede that events. And so we work around a very complicated modeling where we integrate molecular time data, which is basically ability to time large chromosomal gain when they were acquired in the patient life, with structural variants, which is like focal deletion and alterations that occur on this gain. And so again, this can only be done with the whole genome sequencing because structural variants, only whole genome can detect them. And the molecular time can only be run with whole genome sequencing because you need the entire mutational burden, okay? So doing that, we found that in some patients, we have clear evidence of events that were acquired before these initiating events, even before an 11-14 translocation. So it's kind of very interesting for us because it's kind of pointed that myeloma history is not so linear as we think. It's not that there is always the same start. There are different starts, and maybe the same starts can drive to different tumors. And so it's definitely a new area of investigations to understand why a certain tumor with a certain alteration moved toward myeloma or toward lymphoma, for example. We also show that RAG, which is an important mechanism that provides our VDJ or B-cell receptor repertoire in the plasma cells, is not involved in myeloma. So that has been suggested many times in the last 10 years that RAG might be involved in myeloma, in particular in second-1. In our paper, making a cross-comparison with mantle cell lymphoma, where RAG is involved, and with other analysis we perform, we clearly show that RAG is not involved. And so that's, again, it's something important because you need to establish not just when, but also how the events are acquired. So I think this study, it's kind of like very intriguing because it's a kind of sorted the discussion that has been going for a while, but also open up a new avenue of research, say how we can identify these like pluripotent precursor tumor cells.